fast-acting vitamin D receptor agonists for the treatment of IACC conditions, infection-associated chronic conditions. Calcitriol versus paracalcital, Part 3. In this YouTube series, we are discussing three main topics: the nomenclature and structure of fast-acting vitamin D receptor agonists, their medical properties and their indications, as well as the potential repurposing of paracalcital, 1,25 vitamin D2, for the treatment of COVID and long COVID. The goal of utilizing fast-acting vitamin D receptor agonists for IACC conditions is to boost the physiology of remission, and that concept will be discussed in more detail in this YouTube. In part 2 of this series, we discussed these three concepts, highlighted with information vectors. There are two main processes by which currently it is known that vitamin D receptor agonists can boost tissue cell resilience against the impacts of IACC conditions. The first is suppression of ferroptosis, a type of programmed cell death, linked to oxidative stress. A vitamin D receptor agonist, such as calcitriol or paracalcital, can stimulate a vitamin D receptor. This upregulates a master anti-inflammatory antioxidant pathway transcription factor, NRF2. NRF2 can upregulate a key antioxidant enzyme, GPX4, glutathione peroxidase 4, which utilizes the chemical energy and electron from glutathione to chemically reduce reactive oxygen species as well as peroxidated lipids, which are the upstream drivers of ferroptosis. That's the first key pathway which we have been discussing on prior YouTubes on this channel. The second, more recently recognized pathway is the upregulation of an enzyme, CYP2J2. This is a member of the cytochrome P450 family of enzymes. There's 57 of these enzymes in the human genome. This CYP cytochrome P450 enzyme is an epoxygenase, which means it puts epoxy groups on lipids such as arachidonic acid. So CYP2J2 puts epoxy groups on the unsaturated bonds of arachidonic acid. One epoxy group per arachidonic acid molecule. There are 4 unsaturated bonds in arachidonic acid, so the net result is a population of molecules that have varied placements of these epoxy groups. These resulting lipids are known as epoxy-eicosapuraenoic acids, abbreviated EET. They are very unique and very powerful anti-inflammatory mediators, and they are involved in suppressing inflammatory states and prompting tissue healing. For that reason, they are particular importance for helping tissues that have been injured by IACC conditions and the upstream microbial infections to help heal these tissues. In part 2 of this series, we reviewed how paracalcital, 1,25 vitamin D2, can upregulate CYP2J2 to produce EETs. And in this paper, EETs were found to suppress the inflammatory phenotypes known as macrophage M1 phenotypes, pro-inflammatory macrophages. Instead, EETs promote the epigenetic trajectory leading to the M2 polarization of macrophages. These are known as M2 macrophages, and they are involved in suppressing tissue inflammation and prompting tissue regeneration. In this paper, then, it was an animal model, they found that EETs, by suppressing macrophage M1 phenotypes and promoting macrophage M2 phenotypes, that this suppressed diabetic nephropathy, kidney injuries associated with diabetes. And this is the paper which we discussed. Paracalcitol ameliorates, meaning makes less bad, less damage. Paracalcitol ameliorates diabetic nephropathy by promoting EETs and M2 macrophage polarization and inhibiting inflammation by regulating vitamin D receptor CYP2J2 axis. And that is a condensed nomenclature to indicate that there is an interaction between VDR, the vitamin D receptor, and this cytochrome P450 enzyme. The narrative that is encoded in this condensed nomenclature is that the vitamin D receptor upregulates CYP2J2, which has all of these benefits. And that pathway is illustrated in this graphical abstract from the paper. In short, EETs promote the epigenetic trajectory, the formation and accumulation of M2 macrophages, which promote tissue healing. Let us now take the concepts of the previous slide and relate them to the prevalence of autoimmune and IACC conditions. In the next few slides, we will review the prevalence of autoimmune and IACC conditions and relate their treatment to these newly recognized pathways which promote the suppression of inflammatory states and tissue healing. So A and B, a vitamin D receptor agonist, by stimulating a vitamin D receptor, can upregulate the cytochrome P450 enzyme CYP2J2, which acts on arachidonic acid to produce EETs. And once again, EET stands for, it's an abbreviation for epoxyeicosatrienoic acids. These are derivatives of arachidonic acid. They have been epoxygenated by the enzyme CYP2J2. EETs suppress diabetic nephropathy. And diabetic nephropathy suppresses calcitriol biosynthesis. And that's because the proximal tubules of the kidney are the primary site for the synthesis of circulating calcitriol in the body. So in order to understand the impact of inhibiting a pathological process, EETs, by suppressing diabetic nephropathy, help preserve functions of the kidney to synthesize calcitriol. And that's why you see a sequence of inhibitory arrows. This is the way that physiologists, cell biologists, developmental biologists represent a signaling cascade. Some of the arrows are excitatory. Other arrows, the ones with the blunt ends, are inhibitory. It takes a little bit of consideration to understand their significance. One way to think about that is the brakes on a car. So diabetic nephropathy is putting the brakes on the production of calcitriol, 1,25 vitamin D3. If you inhibit the brakes, you then release the wheels. And that would be, in this case, analogous to the production of calcitriol. So slowing the pathogenesis of diabetic nephropathy preserves this critical function of the kidneys to produce this key immune hormone, which is also a key antioxidant and anti-inflammatory hormone. The second information vector shows the impact of a herpes virus, Epstein-Barr virus, abbreviated EBV. Epstein-Barr virus produces a toxin, EBNA3, Epstein-Barr nuclear antigen 3, which directly binds to the vitamin D receptor and inhibits it. So all of the benefits of the vitamin D receptor in suppressing diabetic nephropathy are lost if the vitamin D receptor becomes inhibited by EBNA3, this toxin being produced by the Epstein-Barr virus. So how does this pertain to autoimmune and IAC conditions? Here are 3 papers as background on this subject. The impact of SARS-CoV-2 infection and vaccination on inflammatory arthritis, a cohort study. Epstein-Barr virus as a potentiator of autoimmune diseases, and the information vector encoded in B shows one way that this can occur: the production of a toxin, EBNA3, which inactivates vitamin D receptors, which are needed to promote pathways such as A, the epoxygenase that makes these anti-inflammatory lipids, EETs. That would be lost if the vitamin D receptor has been inactivated by EBNA3. So the utility of utilizing vitamin D receptor-based information vectors is that you can see in a linear code the impact of a pathogen or a pathologic process or the tissue damage that results from these, how they impact the downstream production of these anti-inflammatory hormones. It is incorrect to assume that people who have autoimmune and IAC conditions have fully functional pathways to make calcifediol 25D or calcitriol 125D. This connects to the final reference on this slide, the effects of vitamin D on immune system and anti-inflammatory diseases. A sidebar comment. The use of this term, vitamin D, in the title is very confusing. Vitamin D, the metabolite produced in the human skin, is hormonally inactive. And so it is confusing to read the effects of vitamin D on the immune system and inflammatory diseases. It is the hydroxylated versions of vitamin D which become active hormones. And it's very important to distinguish the state of hydroxylation to understand the impact of vitamin D metabolites, vitamin D hormones, on the immune system and inflammatory diseases. Let us now discuss the potential impact of vitamin D receptor hormones and the stimulation of vitamin D receptors on autoimmune and IAC conditions. Let's start with the graph shown in the upper left, the impact of arthritis on Canadian women. This is published in 2004. Shows the prevalence of arthritis as a function of age. This is a statement from an AI overview of the prevalence of arthritis in American adults. Approximately 53 to 57 million U.S. adults have some form of doctor-diagnosed arthritis, which is 1 in 5, or 21 to 25% of the adult population. Of this, over 1.5 million adults have rheumatoid arthritis, the most common autoimmune inflammatory form. Over 300,000 children in the U.S. also live with juvenile arthritis. It is thought that there are a number of upstream microbial drivers for this autoimmune condition. Let us now discuss this paper, the impact of SARS-CoV-2 infection and vaccination on inflammatory arthritis, a cohort study. Now, this is not new onset arthritis. What they're analyzing is the impact of COVID infections and COVID vaccines on producing flares of inflammatory arthritis. Results: 117 patients and 39 healthy controls were affected by SARS-CoV-2 infection. These are the cohort that were analyzed. 40 patients, 34%, experienced an inflammatory arthritis flare within 1 month of infection, of whom about 7.5% needed to switch their medical therapy. The prevalence of SARS-CoV-2 infection, disease severity, and hospitalization rate were not significantly different. At least one shot of SARS-CoV-2 vaccine was administered in 331 patients and in 147 healthy controls. Within 48 hours, 102 patients developed vaccine-related AEs, adverse effects. 52 patients, that's about 16% of the patients, with more than one vaccine dose experienced an inflammatory arthritis flare-up, of whom 12 needed to switch therapy. That's 23%. Conclusion, a significantly higher rate of inflammatory arthritis flare was observed among patients who contracted SARS-CoV-2 infection versus those without infection. Patients with inflammatory arthritis experienced flares after SARS-CoV-2 vaccination, though it was not statistically significant. This is where prophylactic measures and early interventions with vitamin D hormones, calcifediol, calcitriol, or a synthetic version of calcitriol, paracalcitol, could potentially mitigate the effects of the COVID vaccines in provoking inflammatory arthritis flares. Also, it may also reduce the incidence of inflammatory flares of people with inflammatory arthritis who engender a COVID-2 infection. Now, looking at long COVID, an AI overview. Based on 2022 data from the CDC, approximately 7% of U.S. adults have reported experiencing long COVID at some point. Now, that number could likely be increasing as more and more people develop COVID infections and a subset of them develop long COVID. With about 3.4% reporting current symptoms, rates are higher among women compared to men, showing a significant lingering impact on a portion of the population. So it is known that in COVID and long COVID, there's the reactivation of herpes viruses, which in turn can reactivate dormant fossil viruses known as HERV viruses, human endogenous retroviruses. And so it's not surprising that a major fraction of people who have COVID infections have lingering impacts, symptoms, and pathogenesis and tissue deficits from the transactivation of these viruses. So on other YouTubes in this channel, we have discussed sequential viral transactivations prompted by COVID infections. And we have discussed the potential use of vitamin D receptor agonists to mitigate the impacts of those sequential viral transactivations and perhaps suppress them from occurring. And that brings us to the two papers at the bottom of the slide. Epstein-Barr virus is a potentiator of autoimmune diseases, 2024, a very useful recent review. And the effects of vitamin D on the immune system and inflammatory diseases. Sidebar comment, the use of vitamin D in the title or abstract of papers, or the narrative of the papers, when they're discussing active vitamin D hormones, such as calcifediol and calcitriol, the use of the term vitamin D is very ambiguous. It's a language trap. If you lump all of the metabolites and call them vitamin D, that's not precision language. Let me make an analogy. Adenosine and its metabolites have diverse effects on the body. We do not utilize the term adenosine in the titles or abstract of papers when we're talking about ATP, adenosine triphosphate, or adenosine diphosphate, ADP. And we also distinguish adenosine as a separate player having different physiological effects than ADP or ATP. Adenosine is a sleep chemical in the context of the human serum. Muscular activity during the day releases adenosine to the serum, where it's imported into red blood cells, and then early in the night this adenosine is dumped from red blood cells. Adenosine then binds to adenosine receptors in GABAergic neurons in the brain and brainstem, and it activates them. And when you activate inhibitory neurons, it shuts down neural activity. And that's a first step in terms of transitioning into sleep. So we refer to adenosine as a specific modulator of activity. It's different physiological actions than ADP and ATP. So we need to do the same for vitamin D. It's important to recognize the state of hydroxylation as being as important as phosphorylation is on the adenosine molecule. Now shifting the narrative back to autoimmune conditions. Prevalence of autoimmune and IAC conditions. Approximately 15 million people in the United States, about 4.6 to 8% of the population, live with one or more of over 100 known autoimmune diseases. These chronic, often debilitating conditions disproportionately affect women, who constitute roughly 63 to 80% of cases. Common examples include type 1 diabetes, rheumatoid arthritis, and thyroiditis. In the upper right of this slide, one sees a panel from a paper, Reactive Arthritis Following COVID-19 Vaccination with the Pfizer mRNA Vaccine. This is a severe case of reactive arthritis, which is mediated by immune complexes, which were formed in response to the antigen and the adjuvant that was provided in the COVID-2 vaccine. Could prior treatment with vitamin D receptor agonists, such as paracetamol, reduce the incidence and/or severity of these reactive arthritis side effects of COVID-19? These are severe vaccine injuries. And it's important to acknowledge that these interventions are provoking the immune system, and sometimes the reaction can be very deleterious. In addition, it is known that COVID vaccines can reactivate herpes viruses, which in turn can reactivate HERV viruses, human endogenous retroviruses. So for all of these reasons, it's important to look at the toolbox of vitamin D receptor agonists as a potential way to mitigate the severity and incidence of these vaccine injuries. And this brings us back to the newly recognized pathway by which vitamin D receptors can upregulate anti-inflammatory capacity within cells and tissues. And these involve the production of EETs, epoxy-eicosapentaenoic acids. And once again, these are derivatives of arachidonic acid, which is pro-inflammatory. But when the enzyme CYP2J2 places epoxy groups on the unsaturated bonds of arachidonic acid, it converts arachidonic acid into a family of EETs, and these are anti-inflammatory. Let me now read an AI overview at the bottom of the slide, which connects to the case of reactive arthritis following a COVID-19 vaccine in the upper part of the slide. CYP2J2 is a cytochrome P450 epoxygenase enzyme that produces epoxo-eicosapentaenoic acids, EETs, which have potent anti-inflammatory and vascular protective effects. In the context of arthritis and inflammation, CYP2J2 derived EETs act to reduce inflammation. Its role, namely CYP2J2, its role is studied in conditions involving chronic inflammation, including systemic comorbidities. Now, I'd like to draw your attention to this paper several years ago, which involves an entirely different enzymatic action of CYP2J2. This is not connected with the formation of EETs. Rather, CYP2J2 is involved in converting vitamin D3 into 25-hydroxyvitamin D3, calcifediol. So this is called bioconversion of the vitamin D nutrient or skin metabolite to bioactive calcifediol and calcitriol as high-value compounds, meaning they have important immune functions, including antioxidant and anti-inflammatory activities. So once again, CYP2J2 is a cytochrome P450 enzyme, part of a superfamily of enzymes. There's 57 of them in the human genome. Vitamin D3 bioconversion process. Vitamin D3 is sequentially converted to calcifediol and calcitriol by CYPs, the cytochrome P450 enzymes. Some uncommon CYPs can also catalyze the conversion of vitamin D3 to calcitriol with 1-alpha hydroxyvitamin D3 as the intermediate. Many CYPs, such as CYP27B1, can catalyze hydroxylation at 2 sites, as they have relatively wide substrate specificity and regioselectivity. Putting that in the context of specific types of immune cells, monocytes. Monocytes have the capacity to convert calcifediol to calcitriol using 1-alpha hydroxylase. Now here, they're talking about CYPs converting calcifediol to calcitriol, but the production of calcifediol from vitamin D3 may be prompted locally within the monocyte if it upregulates this CYP2J2 enzyme. And how can that be done? So first of all, it's important to restate that calcitriol, by activating a vitamin D receptor, upregulates NERV2, which upregulates GPX4, which suppresses ferroptosis. So calming the immune system down, reducing pro-inflammatory cytokines, will affect the gene expression within monocytes. Now we add in the impact of calcifediol, or calcitriol, or paracalcitol added therapeutically, how we can stimulate anti-inflammatory lipids, the formation of EETs. And we've seen papers where 125 vitamin D2 paracalcitol, by stimulating vitamin D receptors, upregulates CYP2J2, which converts arachidonic acid to these anti-inflammatory lipids, EETs. So here are key aspects of CYP2J2 in vitamin D metabolism. Role in synthesis. CYP2J2 catalyzes the first activation step, turning vitamin D3 into 25D, calcifediol, which is subsequently converted by CYP27B1 into the active hormone calcitriol. 1,25-dihydroxyvitamin D3, or 1,25-D3, or 1,25-D. All of those abbreviations are found in scientific and clinical literature. Tissue expression of this enzyme. CYP2J2 is highly expressed in the heart, vessel endothelium, liver, kidney, lung, and GI tract. Extrarenal metabolism. This is outside of the kidney, what we've just been talking about. In addition to liver-driven activation, CYP2J2 facilitates local 25-hydroxylation of vitamin D3 and vitamin D2 in extrarenal tissues, and that would include the circulating immune cells, allowing local production of vitamin D metabolites, such as calcitriol. Significance. CYP2J2 contributes to vitamin D homeostasis, primarily when primary metabolic pathways like CYP2R1 are compromised, and CYP2J2 is associated with cardiovascular health. Very pertinent to long COVID as well as other IAC conditions, especially where the vasculature, the endothelium, has been damaged. And it is now known that a hallmark of long COVID is the loss of small diameter capillaries. And this is known as small capillary rarefaction. Regulation. The expression of CYP2J2 and its activity can be reduced by factors like obesity, potentially decreasing local activation of vitamin D. What is local activation of vitamin D? That's hydroxylation. Again, it's very important to specify which hydroxylations are occurring. Hydroxylation at the 25 carbon position creates calcifediol, 25-hydroxyvitamin D3. This is an active hormone. It's about 1,000 times more concentrated in the serum than calcitriol, the kidney metabolite of vitamin D3, 1,25-dihydroxyvitamin D3. Sidebar comment. Obesity is a major risk factor for COVID severity and mortality. This statement about the regulation of CYP2J2 being reduced by obesity, this needs to be examined to see whether the local production of calcifediol and calcitriol is linked to a suppression of CYP2J2 in specific cells and tissues, for instance, circulating monocytes. While CYP27B1 is responsible for the final conversion of calcitriol, CYP2J2 provides necessary 25-hydroxylated substrate. What they're saying is that the production of calcifediol locally within immune cells, and I would add epithelial cells like the corneal epithelium, that provides the substrate which is needed for the cells and tissues to locally produce calcitriol. So in closing, we will reintroduce this paper, which we will discuss in more detail in part 4 of this series. Sequential administration of paracalcitol followed by interleukin-17 blockade for progressive refractory IgA nephropathy patients. They're utilizing a monoclonal antibody to suppress the activity, to neutralize the activity of interleukin-17 in these IgA autoimmune nephropathy patients. The key takeaway from this paper is that paracalcitol is able to boost the estimated glomerular filtration rate, eGFR, in a number of these patients. And it also attenuates progressive autoimmune renal damage. Here is an AI overview of progressive IgA nephropathy. Progressive IgA nephropathy, also known as Buerger's disease, is a chronic immune-mediated kidney disease where abnormal IgA protein deposits in glomeruli. Glomeruli is the plural for glomerulus. In each nephron of the kidney, the first part of the nephron, the filtration apparatus, is known as a glomerulus. There are about 1 million nephrons in a human kidney. That's in a healthy young kidney. With time, the number of functioning nephrons diminishes, and by the time that this kidney function drops to about half, this is well into chronic kidney disease, and elderly patients frequently have depressed estimated glomerular filtration rate, which is a measure of the number of functional nephrons that are present in the kidney. So these steep curves of a diminishing estimated GFR in IgA nephropathy patients, the fact that paracalcitol can boost the eGFR and stabilize the kidney and slow down the progression of this nephron damage, that's what's being shown in the figures in the upper portion of the slide, C and D. We will discuss the details of these measurements in part 4 of this YouTube series. Going back to the AI overview, IgA protein deposits in glomeruli cause inflammation, scarring, and over time a steady decline in kidney function, measured as estimated glomerular filtration rate. This damage often leads to protein and/or blood in urine, which is known as proteinuria, protein in the urine, or hematuria, blood in the urine. Damage to glomeruli, as well as the scarring, can progress to kidney failure, known as end-stage renal disease, in 20 to 50% of cases. So summarizing the results of this paper in a vertical information vector, vitamin D receptor being stimulated by paracalcitol produces renoprotective actions, and that is being demonstrated in this study of IgA nephropathy patients. In the next YouTube in this series, we will connect this paper to the following references: IgA nephropathy secondary to COVID-19 infection, a case report; new onset IgA nephropathy following COVID-19 vaccination; and this review/perspective, the COVID-19 vaccination and IgA nephropathy, further cause for reassurance. In part 4 of this YouTube series, we will discuss how these 4 papers pertain to the question, how does IgA nephropathy pertain to COVID and long COVID? These papers deal with an autoimmune attack on kidneys and resulting kidney deficits, particularly the lack of calcitriol biosynthesis, which will impact the entire body. So how paracalcitol suppresses IgA nephropathy provides key insights to protecting kidneys in particular from COVID and long COVID. So summarizing this YouTube as well as the first 2 YouTubes in this series, stimulation of vitamin D receptors provide 2 major axes that provide anti-inflammatory, antioxidant benefit to organs and tissues. 125 vitamin D, it could be calcitriol or paracalcitol, stimulating a vitamin D receptor, VDR, upregulates the master anti-inflammatory antioxidant transcription factor, NRF2. One of its actions is to upregulate GPX4, glutathione peroxidase 4, which utilizes glutathione to chemically reduce reactive oxygen species as well as peroxidated lipids. The peroxidated lipids are upstream drivers of ferroptosis, And this newly recognized form of programmed cell death is very prevalent in aging, malignancy, and more importantly for these discussions, IACC conditions. The second axis or limb of the vitamin D receptor's benefits comes from a newly recognized pathway, upregulation of a cytochrome P450 enzyme, CYP2J2. This enzyme epoxygenates, puts epoxy groups on arachidonic acid, converting its unsaturated bonds into a epoxy chemical moiety. The resulting molecule is known as an EET, which stands for epoxyeicosatrienoic acid. EETs are able to convert M1 macrophages into M2 macrophages. These are a different phenotype of macrophage. This process of shifting macrophages from an M1 phenotype to an M2 phenotype is called macrophage polarization. It's a term used in immunology to refer to the change in epigenetic trajectory and behavior of these cells. M2 macrophages promote tissue healing. By stimulating macrophages to adopt M2 phenotypes, this helps produce anti-inflammatory activity within a tissue that has been invaded, infiltrated by macrophages. The therapeutic benefits of EETs for the treatment of diabetic nephropathy also has important implications for the treatment of COVID and long COVID, not only for suppressing inflammation and macrophage-induced damage in the kidneys, but other visceral organs as well as the central nervous system. We also began a discussion of a key paper published in 2024 showing that paracalcitol is renoprotective and that in the case of IgA-mediated nephropathy, which is known as Buerger's disease, paracalcitol is able to boost estimated glomerular filtration rate in a number of patients. It also attenuates progressive IgA-mediated autoimmune renal damage. So in closing, I would encourage you to view several other YouTubes on this channel pertaining to ferroptosis being driven by gene silencing. In this case, silencing of the GPX4 gene. And this occurs by hypermethylation of the GPX4 promoter region. And we concentrated on this paper, GPX4 promoter hypermethylation induced by ischemia reperfusion injury regulates hepatocytic ferroptosis. The importance of this paper is that it indicates that ischemic reperfusion injuries, in this case in the liver, are an upstream driver of ferroptosis. It is well established that ischemia followed by reperfusion of the hypoxic tissue results in a surge in production of reactive oxygen species. If the cell does not have adequate antioxidant capacity, for instance, it doesn't have adequate levels of glutathione and or GPX4, there will be a surge in the production of reactive oxygen species, the production of peroxidated lipids, which then trigger this programmed cell death, ferroptosis. And this can occur potentially in other tissues as well. For COVID and long COVID, this is very pertinent because, especially in long COVID patients, many of them are afflicted by numerous microclots that damage microvasculature. And ischemia reperfusion injury is a serious consequence of trying to dissolve these microclots without prior boosting of the tissue's antioxidant capacities. And that's where these vitamin D receptor agonists could potentially be used beforehand, before microclots are dissolved or dislodged by therapeutic approaches. If you would like a PDF slide summary of this particular YouTube, please go to the Google Docs folder highlighted in green at the bottom of the slide. Here you will find a set of subfolders. If you'll navigate to the subfolder Download First, you'll be able to find a PDF slide summary of this talk more quickly. There's also an archive of other PDF slide summaries of YouTubes on this channel. Also on this Google Docs folder are a set of subfolders that contain pertinent references to topics such as COVID and ferroptosis. You're welcome to download all of the material from this Google Talks website. And we will now proceed on to the fourth part of this YouTube series.